Yes, I am Dr. Jayakumari, working as professor, Department of Pharmacognosy, School of Pharmaceutical Sciences, Vistas. Today I am going to take about the Unit 3, carbohydrate containing, organized drug containing carbohydrate, under that one cultivation, collection, preparation, various uses, Isapgol. It is synonym Isbagla, Indian psyllium, flea seed, Plantago. Isapgol is a dried seeds of the Plantago ovata family Plantagenaceae. They are cultivated in Gujarat, Punjab, South Rajasthan, Maharashtra. Coming to the plant habitat, it is about annual plant. It grows very well in the climatic condition, cool, dry weather, altitude 1400 1008 meter, temperature 30 to 35 degree. It require rainfall 40 to 16 and needs well drained loamy soil, cool and dry, cultivated by broadcasting method. It was propagated by broadcasting method. Type of propagation broadcasting method, time of collection November. The field is well irrigated. Field is well irrigated 7 to 8 days. Ammonium sulfate is added as a fertilizer for about 4 months after harvest. The harvest can be done after 4 months from the matured seeds. After 120 days, the mature seeds are obtained. The seeds are harvested in April and May by cutting above the ground level, then converting into seeds. Then they are dried in a shade, and then they are thrashing is done, and the winnowing process is used to separate the seed. Finally, the seeds are separated from the impurities by sieving method. India is producing 48,000 tons of seeds annually at present. Coming to the Isapgol husk, these are the outer seed coat of the Isapgol seed. They are crushed in flat stone, grind stone by passing several times through them to remove the coat completely. Then it is separated by winnowing process from the seeds. The kernels, separated kernels are again sheaved to get rid of the different grades and different sizes. That it consists of the epidermis and its adjacent layers removed from the outer part of the seed. It is free from pale buff ovate flakes and lanceolate shape. Coming to the macroscopical character, the color of the seed pink is gray brown, odorless, mucilaginous stage, size about 10 to 30 millimeter long, 1 millimeter wide. It is shape is somewhat lanceolate shape or oblong shape, boat shape in nature. There are two views we can see, dorsal view, ventral view. In the dorsal view, we can see the center, there is a hilum will be presented. In the ventral view, there is a membranous structure will be presented. It represents the hilum. Coming to the microscopical character of the seed, the outermost part of the seed is a testa, then followed by the endosperm and embryo. The testa outermost layer contain epidermal layer, somewhat irregularly flattened structure, followed by the pigment layer. The testa of the outermost layer contain mucilaginous cell. This mucilage gives us a red color with ruthenium red dye. Endosperm is completely packed cell without intercellular space. It contains oil cells and alluron grains. Endosperm cells also containing some grains, starch grains, and some of the crystals are presented. The outermost layer of the epidermis contains mucilage layer. Seeds are hard, transparent, smooth with gray and reddish brown oval spot in the center of the convex surface. Concave surface contains hilum covered with a thin membrane having two perforation which accommodates about 500 to 600 seeds. Coming to the chemical concept, the seeds mostly contain carbohydrate. The main carbohydrate is a polysaccharide mucilage. It is chemically made up of pentose and aldobionic acid. With the product upon hydrolysis gives xylose, arabinose, galacto-uronic acid, amnose. The seed also contain fixed oil, proteins, other important constituents of the drug. Coming to the important parameter, physical parameter for identification on the seed is a swelling factor. It is a quantitative estimation of the swelling due to mucilage present in the drug. It was determined by taking 1 gram of the seed in 20 ml of water with occasional shaking in 25 ml measuring cylinder. The volume occupied after 24 hours is measured. That indicates about 10 to 14 ml. It indicates swelling factor. It is a quantitative value of the mucilage content. By chemically, the mucilage was tested by treating the seeds with the ruthenium red. It gives us a pink color, which is due to presence of mucilage. Coming to the uses, because of the mucilage, it is used as a demulsion, bulk laxative, emollient, the treatment of chronic constipation. It also used as a bacillary and amoebic dysentery as a emollient property, demulsion property. Mucilage is also used in the preparation of tablet binding agent, stabilizing agent in ice cream industry. The crushed seeds are used in the form of poultice for the treatment of the inflammatory pain, that is rheumatic pain. The acid form of the polysaccharide of the seed is used in entry coated tablet and tablet disintegrating agent and sustained release formulation as a disintegrating agents. These are the some of the substitute: Plantago rhodosperma, Plantago rhytinia, Plantago cruzi, and Presto aristata, asiatica, and Celia. These substitutes can be evaluated by swelling factor and chemical tests. Adulterants, the one of the adulterant very commonly used is a lanceolata. It is a lanceolate in shape. The seeds are oblong, elliptical in shape, yellowish brown in color. Swelling factor is about 5. Swelling factor is a drug quantitative measurement of the swelling due to mucilage present in the drug. It is a criteria for determining the quantity of the seed present in the class as well as determining the quality of the seed also. It was determined by the simple method. Take 1 gram of seed, 25 ml cylinder, 20 ml of water was added. The cylinder is shaken very frequently for about 24 hours. The seeds are allowed to settle for 12 hours. The total volume occupied by the seed was noted. The seed of Plantago ovata occupied about 14 ml. The swelling factor of psyllium is about 12.75 ml, which is less than that of the ovata, which comes from the that one. Plantago lancilla has a very low swelling factor, about 4 to 25. That is, it is very commonly used as an adult drug. This is a apparatus used normally. It is a 25 ml measuring cylinder. It is a initial volume seed occupied by the water. Then after that 12 hours over, that seed volume was increased, whereas the water volume was decreased. It was noted as a final volume. These are some of the commercially available Isapgol products in the commercial market for the use of the laxative property as well as the nutrient property also. These are the references I referred for this slide. Thank you, students.